to make sure that we can balance the nutrition in our soil, okay? And hopefully, by doing that and having a biologically active soil, it will eventually get into the plant, right? Which of these do we need to design for a good program? All of them. Unfortunately, a lot of people, I work with a lot of conventional farmers as well, sustainable and true organic farmers. Conventional guys, I mean, they're only looking at chemical. That's all they're doing. Unfortunately, you got to bring them across. And a lot of guys that I work with are sustainable, and they really don't realize what they're doing. But they see the production. They see the benefits of using biological products, using compost teas. Okay. So, how do you help prepare a fertilizer recommendation? Soil test, okay? Anything is better than a guess. So I highly recommend start with a soil test. This is your baseline, okay? What it does, it identifies yield-limiting nutrients, okay? We got to make sure we ascertain what we're starting off with, okay? On top of that, it will also indicate nutrient supplying capacity. How much can it hold? Going back, you hear Neil Kinsey talking all the time about count-on-chain capacity, okay? Lower count-on-chain capacity, the less ability for it to hold nutrients, unlike a clay soil, higher. So we got to make sure and manage that properly. Provides information for nutrient management. If you're using manures, compost, and so forth, we got to make sure that we keep that system balanced, not overdo it, okay? I work with a lot of dairies that use the excess manures on there. Number one problem, excess nutrients. We got to balance that out to minimize a lot of the stress. Okay? Monitor the production system, keep records of nutrient management, you know. You got to keep all that data and make sure that you provide this data to your crop consultant as well as, or you're working with your laboratories as well. Okay? Bottom line, all this information will help manage risk because that's what we're trying to do. And stress is a way to do it. Minimize stress. So you've probably seen these nutrient requirements for plant life. There's what, 21, 20 up there. Generally, you'll see some people say 17, 18. And you've seen some up there that you might not have seen before. So basic nutrients, generally available for plants in sufficient quantities coming from the air, through the soil, or from the irrigation water. Okay. Unfortunately, we're seeing more and more where we've abused our soils over time, over tillage and so forth, where the carbon sources have diminished over time. So how can we do? What can we do? Cover crops, compost, that helps. But how does that contribute to stress on your plants? Okay, think about that. Hydrogen, oxygen as well. Now you look at your major or primary micronutrients, nitrogen, phosphorus, potassium, the big three, you know. Mostly the focus of your traditional quote-unquote program. Again, going back to my conventional guys, traditional nowadays, unfortunately, is nitrogen only. And that's the worst thing you can do when you're growing a crop, is just applying nitrogen only. Okay? Secondary nutrients, calcium, magnesium, those are the bases. You've got to make sure and build that base before you can build upon it. And you'd be surprised how many of these calcium, magnesium, sulfur deficiencies there are throughout the entire U.S., causing a lot of stress. Okay? Micronutrients, iron, manganese, zinc, copper, boron, moly. These are generally not the primary focus of your quote-unquote traditional program, but they're vital. And these are the ones that are key to mitigating a lot of stress in the plants. Okay? Unfortunately, the data out there stipulates that it's only required for plant reproduction, early vegetative stage to initial reproductive stage. But health-wise, they're essential throughout reproduction, late into the season. And that's where a lot of people stop short. They apply starter fertilizers, apply these micronutrients, which is essential, and then forget about them. And that's what causes stress. Those deficiencies mid and late season. And I'll show you what these do at these key stages. Okay? Some of these other ones that are generally not called essential, and few people or few labs actually test in the plant as well as in the soil: cobalt, selenium, nickel, and silicon. Silicon is one of my the ones I've been doing our own research in a lot of crops, and I've got some more details on that here. So this is some information from International Plant Nutrition Institute. Highly recommend you guys looking at the data. So basically what they've done is they take the ag census data for down to the county level and extrapolated the amount of fertilizers applied, the amount of compost, the amount of manures applied, of course, taking away the yield response, or the yields taken off those information. So you can see from 1987 to 2007, 3 major nutrients, N and P, climbing a deficit of 35%, with potassium climbing about 26%, okay? Now this happened while the acres declined from 443 million down to 410 million in 2007. So what that means is that the nutrient removal to use actually applied is very high. We are removing way more nutrients that we're actually applying. And that's what's causing stress. They have a website they call NUGIS, stands for Nutrient Use Geographic Information Systems. Now this is a particular map, deficit map for potassium. And it's hard to see, but this is 1987. So if you look here at the legend here, your oranges to reds are indicating a deficit, okay? So you can see the primary western part of the U.S. here is it has a potassium deficit. Upper East Coast, low. But look what happens when you jump to 2007. Major part of the U.S. now showing major potassium deficiencies, okay? Look at the eastern part. So that's where you see a lot of the stress coming in. Unfortunately, a lot of people use hardly any potassium or very small amounts of potassium. In fact, I've been talking to some researchers the last 10 years that they're getting a yield response to potassium additions, even though their regional soil tests say they got plenty of potassium in their soil. Okay. So now we know that we've got a deficit, okay? What can we do? So there's some data here. So to rebuild the soil, when it comes to phosphorus, it takes 10 to 15 pounds above the removal rate. So let's say corn removes 80 pounds of phosphorus. We got to have 80 pounds there, plus an additional 10 to 15 pounds on top of that, just to increase 1 pound in your soil test to rebuild that. Cost effective? Probably not. But if you start incorporating cover crops, start incorporating manures, compost, it's feasible. Look at potassium. 5 to 10 pounds above the K removed by the crop to raise the soil test by 1 pound. So I tell people when you're considering going organic and you're taking over a farm that's been highly harvested for many, many years, do a soil test, see where you're at, but consider at least a 5 to 10 year approach to rebuild those nutrients, okay? In fact, a lot of the initial people are thinking going organics. I sometimes tell them, let's go sustainable so we can sort of cut back on some of that time needed to rebuild the soil, then stop all conventional fertilizers and do organic just to make it more cost effective. But some people, they just say, you know, I want to go organic and strictly, but that's the face of reality. At least 5 to 10 years to rebuild it when you're taking over an operation like that. So plant analysis, soil testing go hand in hand. So what's the difference? Okay, so soil tests and plant analysis are complementary. So soil test is a prediction, you know. This is what I call the knowing versus hoping, okay? Where when we apply fertilizer to the soil, we're hoping those nutrients get into the plant, right? Because there's no guarantee when you apply that fertilizer, whether it be organic or conventional, it will get into the plant. No guarantees. Now, if you use plant analysis and you know how to interpret properly, that will assess the nutrient uptake. And that will tell you if that fertilizer applied is getting into the plant when it requires it. Okay? Now, not all soil tests are good predictors of nutrients. You know, for example, nitrogen and sulfur, they change depending on time of year. Okay? Now, plant analysis as well. Some of those micronutrients are more better evaluation of, you had a question, sir?
Yeah, sorry. So the soil test, do you think that that is testing everything that's available, like so phosphorus or something that gets tied up in the soil? When I have a soil test and it tells me I have this level, is that all readily available for the plant, or is that just something that's mineralized and tied up in the soil that I can't get at?
So the question was if the test that he's getting boron is that's available portion or the total. It depends on the laboratory and the extraction they used. There are some harsh extractions, acid extractions, that give you a total. Now there's a percentage in there that's available, but you don't know what percentage is. Now if you use a method like hot water, that is available boron, okay? But when did you do the test? If you do it in the fall, hoping that will be in spring,
Yeah.
and you've got a very low organic matter content, remember boron is an anion, so it'll leach away. Most likely if you got a wet winter, might not be there when you require it. So there's different aspects you got to consider when looking at that, okay? So basically, what we want to do is use plant analysis and identify the hidden hunger, okay? What we want to do is, before we see those visual deficiencies, act on it, okay? And apply the necessary nutrients required, because by the time you see those visual deficiencies, you're already at deficit and it's already costing you production, okay? So if you use this tool as a proactive approach rather than a reactive approach, it'll more than pay for itself. And that's the way, and that's the key to finding this maximum production. Okay? So in most crops, yield potential is set the first half of the growth cycle during the vegetative growth stage. Very early. Okay? Attainment of that potential is set second half of the reproductive stage. Okay? So, crop, V5 stage, 20-25 days, approximately day after planting, potential seed form is formed on that cob. 20-25 days. So what you want to do is you want to test before V5 stage. V3 stage, V2 stage, pick the whole plant, and let's see before you get the genetic potential. Unfortunately, a lot of consultants, they do their first test close to taxing. Well, that's kind of late, guys. If you're setting your yield potential at V5 stage, why are you testing way too late? You can't make any more changes to the genetic potential of that plant. You got to start early. Wheat, around phase 2, right before tillering. Why do you wait until boot to take your first tissue test or leaf analysis, sap analysis? Start early. You want to maximize the tillering on that wheat plant. Why? Because that is going to promote your number of heads and increase your yields, right? Soybeans, V2 to V3 stage, 20, 25, 30 days after planting, potential nodes for increased blooms. Yet, you got some people who are recommending taking your first sap analysis at bloom or pre-bloom. Way too late already, because genetic potential set very early, V2, V3 stage. So that's an embryo, V5 stage, to the embryo. Zinc is essential at this stage. Okay? If you don't have zinc, then that will limit the number of rows on that head, on that cob. Zinc at that stage dictates whether you got 16, 18, or 20 rows on that cob. So you got to make sure that your hidden hunger is identified right at V2, V3 stage. Make sure we have enough. Now, people say, well, we can use a pop-up fertilizer and hope that it'll get into the, exactly. You can hope it gets there, but there's no guarantee it'll get into the plant, okay? Plants don't lie. That's what we call ask the plant. Let the plant tell you whether it's got enough. If it's not, we'll have to come back and do a foliar spray. Maybe we get lucky, we get the fertilizer, proper adjuvants in the soil, your pH isn't as high, and it gets in the plant, and we're okay. We don't have to worry about adding any additional zinc to meet genetic potential of corn. Okay? So plant analysis, again, take a very representative sample. Just like soil test, I tell people take a representative composite sample of the entire growing area, keep it with the same variety, keep it same leaf, same location, same stage of row, and most laboratories will provide ways or which one to take. So which leaf to take is very important. Make sure you get with the laboratory consultant which one to take. because they all taste different. You get too young, it's going to be way different than an older leaf. Rule of thumb, you always want the most recent, fully expanded leaf, okay, on any crop. Sample timing, start early, okay? And I just showed you what the genetic potential of a lot of these crops are. The earlier you start, do a whole plant analysis, the better you are off, okay? Not only yield potential, but also mitigating a lot of the stress. Bottom line, we've got to eliminate stress. And I'll show you some of the stress problems. The other thing, too, is the interpretation of that data. What does that data mean, okay? And I'll tell you what some of the issues that I found out there when we work with a lot of farmers and laboratories. And ultimately, recommendations. What products do we use to address these deficiencies? And there's a lot of wonderful products. Talk to your consultants, look into your lab guys and so forth, see what they recommend. So leaf analysis, what's the difference between leaf analysis and sap analysis, okay? So leaf analysis, basically we call it postmodern autopsy. It's an accumulation of nutrients that have occurred in a plant from initial emergence to the day you take that leaf, okay? Is it good data? Yes, it is. Very good data if you know how to interpret that data properly, okay? Now, on the other hand, sap analysis, that's what I was talking to being a proactive approach and identifying the hidden hunger before you see the visual deficiencies, okay? It will forecast up to 21 days in advance what the plant will be deficient in before you see them on your crops, okay? So this is the general nutrient and water use curve for all crops. So planting, early vegetative growth, you notice at very low demand. Once you start into a reproductive stage around bloom seed field, the demand increases, not only for nutrients, but for water as well. But again, stress levels have to be minimized early on. And then towards harvest, of course, that demand decreases. Those are typical ranges that you'll get from laboratories and so forth on most leaf analysis. Now, some of this data dates back over 50 years, okay? That's your macro and your micronutrients, boron, copper. So that's what they consider sufficiency ranges, okay? So, plant analysis results are compared to these establishing ranges, right? That's how we can let you know whether you're low, medium, or high, okay? Unfortunately, a lot of these laboratories are still using standards that were developed in the 1970s. So what am I getting to here? Well, how can the newer varieties be interpreted or identified whether they got deficiency or not with 50-year-old standards? Even worse is, if you want to go after higher yielding capacity, those old standards were based on much lower unproductive varieties. Let's take corn. What does that data come off of? Maybe 150 bushel corn? But what if you're going after 250 bushel corn? Can you use the same interpretive guide to identify the deficiencies? You cannot. So let's take this analysis, for example, and let's take a look at potassium. So here, potassium, 2.6%. And that laboratory gave you a acceptable range of 2.4 to 4%, okay? I know, everybody sees that. Well, I'm within range, right? Above 2.4, 2.6, I'm fine. Well, what stage of growth was this plant taking? So let's assume for all purposes that we are around close to tassel. But look at the amount of potassium close to tassel, very high. So if you're at that bottom end of that acceptable range, do you think you'll have enough potassium for that crop? You will not. You will not. And that's the reason I'm talking about. We've got to adjust these plant standards to identify them at these key potential physiological stages of growth, okay? We've got to make sure we interpret the data properly and we're interpreting it appropriately to the yield potential we're having. So if you're looking at 250 bushel corn, but your standards are for 150 bushel corn, that interpretation is invalid. And that's the reason why you're getting responses to potassium additions, even though soil tests come back, you got plenty in your soil. You've seen these nutrient deficient symptoms, you know, your micronutrients, your new leaf deficient or chlorotic, generally your trace elements. Your older leaves are chlorotic, generally your macronutrients. Again, these visual deficiencies, they're great, anybody can do it, but what is it costing your production? How much stress is doing onto your crops to mitigate a lot of risk management we got to do? So that's typical nitrogen deficiency in most plants. There's some corn that leaf that starts turning brown, generally less than 2%. Now this is some data from the University Extension. Up here, this is for 180 bushel corn. So look at the nutrient usage for nitrogen here. First 25 days. Only 8% of the total nutrient usage is required first 25 days. But look, once you get into rapid growth, 25 to 50 days, 35, 30% starts increasing. So that's the problem that a lot of farmers have. They put all their nitrogen early on when the need is very minimal, and you get this very vegetative plant, susceptible to disease, insects, weed pressure. Not only that, is that it's very difficult to a plant to go or transition from the vegetative stage to reproductive stage because of that excess luxury consumption. And that's affecting stress. That's affecting your yield potential, okay? Wheat, same thing. Look, first 30 days before tillering, only 24%. But look, when you get into reproductive stage here, 44% of the total in. Same thing for phosphorus, 7%. So all your major nutrients are minimal used first 25, 30 days after emergence. So when you put all that fertilizer up front, guess what? Causing undue stress. So if you can spoon feed that plant, if possible, that's the ideal way of doing it. Not sure if many of you guys have seen cotton, but that's cotton, and that should only be about waist high. That's too much of a good thing, too much nitrogen, and again, it's a multi-fruiting crop. Way too much nitrogen put it on too early and causing that excess vegetative growth.
He wasn't kneeling down, was he?
No, he's not. And if there's any Aggies in the crowd, I apologize. He was wearing an Aggie cap. I get grief for that sometimes when I show that slide. Phosphorus deficiency. It's funny, I got a call this year. One of my consultants say, hey, you know, I got some corn that's turning purple. Talk to the seed company. Seed company say, oh, it's okay. It'll grow out of it. Well, yes, it may, but what stress is it causing the plant? Okay, that early on. What if that stress comes in at B5 stage when genetic potential is being set? What does that do to your crop? Okay, so again, plant analysis will help you circumvent that. So phosphate, I call it the root activity indicator. It will tell you how productive your roots are. It will tell you how especially your young feeder roots are. Because that's key. You want to keep those young feeder roots as active as possible, okay? Once you get those older roots, they start sloughing off, and the uptake of phosphorus is diminished, okay? So this is some data that we did. This is on watermelons, but this applies to all crops. So there was 10 weeks, if you notice, right after harvest, we're at about 3,700 parts per million of phosphorus. Ideal, we want to see it around 3,500. But look what happened. All of a sudden, it just dropped. Okay? So on plot 1, we didn't add anything. All it had was that early pre-plant phosphorus. Okay? And it just dropped. So number 2 section, this was the drip irrigated watermelons. Number 2, we added a high P formula. And it just still well below the 3,500 parts per million. So in plot 3, all we added was a humic fulvic acid product. And look what happened. Without adding any additional phosphorus, it went higher than a phosphorus addition. Now on number 4 plot, we added phosphorus plus a humic acid fulvic acid combo, and it gave it a synergistic effect. Remember, humates are natural complexing agent. It increases the uptake of nutrients, especially phosphorus. So in number 5, what we did is added the phosphorus, humic fulvic, but we added hormones, seaweed or kelp. They're laced with a lot of good hormones in there. Look what it did. It almost getting us to 3,500. So at the end, we added phosphorus, humic acid, hormones, and these beneficial microbes, the soil and alkalines everybody's talking about, or compost teas. Now you got us well over the 3,500 parts per million, okay, where we need to be so we can get more fruit set. But look what happened to the nitrates without adding any additional nitrogen whatsoever. Every time we increased that phosphorus, remember root activity, those young feeder roots, it enhanced nitrogen uptake. And that's key. That's how you can maximize genetic potential while keeping your input costs down without adding to have any additional nitrogen whatsoever. Yes, ma'am?
Is that actual nitrate levels in the sap?
That's,
Or is that nitrogen converted to equivalent?
no, it's nitrate in the sap. Yes?
What was treatment 0?
Treatment 0, that was at harvest. So right after harvest, all of a sudden, our values dropped. Okay? So in a multi-fruited crop, we got to keep that plant as productive, reproductive as possible. If we would have kept it there, they would have only a single cut. When we got this program going, we're dosing this, we kept those young feeder roots, we can have 3, 4, 5 cuts in the season, depending on the market.
So are you applying foliarly or to the soil?
This was injected through the drip irrigation, subsurface drip irrigation. Yes, sir?
Have you found any problems with too much phosphorus? Because I've got a ton of phosphorus in the soil,
Yeah.
and I've never really seen anything bad about it.
Yeah, question was, is if you've seen any issues with excess phosphorus? Yeah, you induce other deficiencies, specifically zinc. So in your case, you know, I'm talking about genetic potential of zinc early on. If you got high phosphorus, guess what? You'll induce zinc deficiency, even though your soil tests come back plenty of zinc in the soil, but it will not get into the plant because you're inducing deficiency due to the excess phosphorus. So you got to balance that out.
It's bonding with it and then tying it up so I can't use it?
Yes, yeah, it will induce it, and it will not get into the plant. So again, you've got to know how to interpret those results. Even though the soil test says there's plenty of zinc, it will induce a zinc deficiency. Dairies that use all that manure, very high phosphorus levels, and I see that. The other thing too is that dairies that are using the hoof wash with copper, excess copper will induce iron deficiencies. So you've got to balance all that out.
Are those reversible at all? Like once it's tied up, is there anything I can do or apply or anything?
You'll have to apply to get that balanced out properly because there's nothing you can do to the soil to prevent it. Okay? Potassium deficiencies, again, typically seen in the older leaf, bottom leaf. You get the inverted V on that leaf. And generally that happens because it's translocating from the older leaf to the young leaf, thus senescing. Okay? Folks, we got to keep those leaves on this plant as long as possible. Because remember, they're the ones collecting solar radiation, converting carbohydrates, and setting your fruit. Every time you lose a leaf, guess what? You're losing that carbohydrate factory. So you got to keep those leaves as long as possible green, working for you. Okay? Again, you never want to see this browning at tassel. This is R3 already pollinated, but you want to keep those as green as possible. You don't want to see that. This is some sugar beets. I was started working about 5-6 years ago. I was driving up there, and they were wilted right at 2 o'clock in the afternoon. Say, oh, that's common. I said, well, how much stress is that causing to your crop? We did a 1-to-1 ratio, same amount of potassium to N. Never had that issue again. Yes, sir?
Was that foliar applied?
Yes, we ran around that through the pivot. So the question was if the potassium was foliar applied, and we ran that through the pivot. Okay? Sulfur. We're seeing a lot of sulfur deficiencies. Constitutes of proteins. Increases protein in your forages and grain. People assume that we've got to add all this nitrogen, increase your proteins. That's not the case. You do undue stress in the plant. It actually decreases over time the protein and increases your nitrates, which you don't want to do. You want to make sure and have sulfur and balance that out properly to increase your proteins. Again, avoids nitrate buildup in forage crops, involved respiration nodule formation in legumes. The other thing too is a lot of times, a lot of labs are looking at sulfur recommendations as a nutrient uptake. I like to approach it as a soil condition as well. Because remember, we talked about that three-legged stool. Physical aspects of the soil is very important. Chemistry can help you with the physical contact of the soil. You got to make sure that we have enough soluble calcium, magnesium, so we can increase the soil till, so we can have more aerobic conditions, and sulfur helps you with that. Okay. Calcium, you hear Neil Kinsey talking about this all the time. The cell walls formation. It's a foundation. It is vital to get into the plant. Okay. Aids in translocation of carbohydrates. You heard me talk about potassium. Well, calcium sort of helps increase potassium deficiency, okay? But the same token is you get too much potassium early on during the vegetative growth, guess what happens? You induce a calcium deficiency, okay? So it's all about balancing, okay? It increases fruit set as well, stimulates microbial activity. Why? Because once we get enough calcium in the soil, soluble portion, at least 150, 200 parts per million, now that increases the soil till. We increase the soil till, now we got more aerobic conditions, more oxygen getting to the roots, okay? From now on, you start seeing this bottom line, you'll see about these key nutrient cofactors. You hear me talk about adding hormones, seaweeds, kelp, and so forth. Well, the plants can generate its own hormones, okay? But they've got to have enough of these trace elements for the plant to generate themselves or secondary nutrients. So you're not only getting the benefit of the calcium for cell wall production, you're also getting the benefit for increased production of these phytohormones that are essential for plant reproduction and increased yields and less stress. The more that balance of hormones are, less stress on the plant, okay? So we either add it or we can manage it nutritionally. Make sure that we allow the plant enough balanced nutrition so it can reproduce its own phytohormones. Magnesium, vital as nucleus of the chlorophyll formation, okay, for photosynthesis. Essential for sugar and starch production. Aids in phosphorus metabolism. I've been seeing a lot of magnesium deficiencies, and you'll see it typically in corn, that yellow striping on corn, the older leaves and so forth. That's typical magnesium deficiency. Again, you got to be careful. You got to balance that out. We got to make sure and avoid that early on, because remember, once you get into the R1 stage, close to pollination, we got to make sure we got enough magnesium. Unfortunately, potassium, magnesium, they work against each other, just like potassium and calcium. You get too much potassium early on, you induce magnesium deficiency. And you'll see it. You look enough lab data on sap analysis, early on you're always going to see higher levels of potassium and lower levels of magnesium. Once you get into the latter part of the season, you'll see magnesium increase and potassium decrease. Okay? So we got to learn how to manage that properly. Okay? Aids for respiration. Again, key nutrient for cytokinin, auxin and gibberellins, those hormones I was talking about. For cattle, causes grass tetany. So very common. Make sure we can address that magnesium deficiencies. Zinc, again, essential for several important enzyme and growth hormone systems in plants. Necessary for starch formation. Essential for maximizing genetic potential. And this applies for most crops. Again, you really need to have that early on in the vegetative stage of growth, 20, 25 days after emergence. You got to make sure you got enough zinc in the plant, okay? Can you overdo it? Yes, you can. So you got to make sure and monitor it, and that's where it's key to do a plant analysis and ask it, do we have enough? Do we put too much? Did we not put enough? Did we use the wrong chelation, okay? Do we use enough in our pre-plant to get into the plant when we require it? So you got to ask yourself all these questions, okay? Again, key nutrient factors for those micronutrients. For cattle, calves are shy in both immune response and muscle growth. Typical signs include reduced feed intake and skin lesions when you have zinc deficiencies. Iron, essential formation of chlorophyll and photosynthesis and a lot of these different enzymes in the plant. Important for respiration and oxidation systems in the plant. Iron is one of these nutrients that have seen a lot of difficult deficiencies. Unfortunately, it's difficult for consultants or any farmer to be looking at a plant, assume it's always iron. Okay, it could be zinc, it could be manganese. So that's why you got to ask the plant and figure out what it is. But a lot of the irons that I've seen is generally due to either lack of soil moisture or excess water moisture. I like to take a look at iron as my key to let me know how my irrigation is. If you're overwatering, you will see an excess of iron in your sap analysis. You're talking over 250 parts per million. Sometimes you'll get 500 parts per million. And that's a key indicator that you are overwatering. And the reason that happens is because iron accumulates in the sap because of anaerobic conditions. You don't have enough oxygen going to the roots. Vice versa, if you're too dry, the iron in the sap will drop. So that's another way to use to help you manage your irrigation system if you are irrigating. Key new factors for cytokine and Auxin and Gibberellins. Of course, for cattle, primarily requires the formation of hemoglobin. Signs for include anemia, depressed immunity, and decreased weight gains. Manganese, essential part of several important roles. And Don Huber talks about this all the time in Roundup Ready crops, manganese. But I'm not only seeing manganese in a lot of these crops, I'm seeing magnesium, I'm looking at zinc, I'm looking at boron. It is more than just manganese. It's a combination. I think it's cascading effect that's happening. And I said I work with a lot of these crops, Roundup Ready crops, and every time we start seeing that first foliar spray come on, manganese, zinc, copper, magnesium, all drop. Why? And Don Huber stumbled on it, and he's telling us what's happening. Problem is this is happening when the genetic potential of the crop is being set. So you can see how counterproductive that is. So what do? Do we delay that foliar spray? And that's what I told my guys. Or avoid it if possible. Or we can come back and do a foliar spray and circumvent that. And that's what we're doing. But a lot of people don't realize that. For years, we were looking at it in my mentor Kate Chandler. We talked about it. We see this sharp decline in micronutrients. We never knew what it was. And so I started talking and listening to Don Huber and started seeing all that data go back, and it makes perfect sense now. Okay. Copper, again, serves as an activator of several plant enzymes and plays a role in vitamin A production. Deficiencies interfere with protein synthesis. Reproductive stages, essential for disease suppression. I see a lot of copper deficiencies in a lot of crops that I grow. Generally, we want to see levels around 10 parts per million. One of the major signs in cattle that I see is that rusty appearance of black-hided cattle. That's typical copper deficiency. But again, what stress is that causing to your cattle? We want to avoid that stress. Well, we can provide these minerals, free choice minerals, right? But that's not the way it was intended. Why don't we provide this to our grass and let the grass, the way nature intended, feed our cattle? And then you've got much less stress on the cattle and the crops. Boron, major functions, differentiations of meristematic cells. I consider boron the cake keeper. Without boron, guess what? Calcium uptake, silicon uptake, all these trace elements will be very, very low. We have to increase that. And boron is one of these bad omens that a lot of people have. Well, you know, you hear about boron toxicity and so forth. Since my career, I've only seen probably 2 boron toxicity events in my life. And that had nothing to do with any application. But every time we increase the boron, we see an increase of reproduction, we see an increase of yield, we see less stress on the plant, okay? Molybdenum is another major deficiency in a lot of crops. I tell every time I work with soybean growers, pull up the roots, cut up that root nodule, check the color. It's supposed to be that red brownish color. If it is white, it is not fixing any nitrogen whatsoever. I had some guys that done right at bloom. We tested moly in the sap, it was very low. We did an application, probably cost $7 an acre, had an increase of 15 bushels with a single application of moly. So it's very, very cost effective if you know what you're looking for, if you're asking the right questions. Most labs will not provide that data unless you ask for it. Or very many, they assume, well, we applied it with a rhizobia, with an Auxin, it's in the plant. Not necessarily. There's a lot of induced deficiencies that occur. Chemistry occurs in the plant, and the soil as well. Okay. Cobalt, sensor for nitrogen-fixing bacteria as well, for legumes. Cobalt again. And that's probably why we see a lot of these effects when we use seawater. Okay? Because it is very rich in a lot of these trace elements that are generally not typically in your quote-unquote traditional program, right? You know, when will you find a conventional fertilizer and read the label and find cobalt, moly, selenium, silicon? Unless you really look hard and look for it, you'll find it. Otherwise, it's very difficult to find. And that's why you see a lot of these increased uptake, a lot of these increased yields, higher Brix levels when you apply these sea salts and so forth, because of these other trace elements that are in that product. Selenium helps with the tolerance of UV-induced oxidative stress. You know, I was talking to a gentleman here a few days ago talking about lavender, where he says we're seeing a lot of stress on those plants where we got that sun. Again, I told him, let's look at selenium. It really helps. In fact, UC Davis is doing some studies on selenium as well as abscisic acid. Why? Because it helps regulate the stomata, just like potassium. Really keeps your evapotranspiration rates down. What happens when you got very high ET levels? When the plant gets down with it, it's got to use its enzyme production at the cost of using carbohydrates. So instead of using that carbohydrate for reproduction, it's using it to heal itself. But if we can minimize that evapotranspiration rate, minimize that stress by using selenium or increasing abscisic acid, then that won't be the case. And you minimize the stress on that plant. Okay? Again, it regulates water stress, the status of the plants during drought conditions. In cattle, retained placentas after calving and weak calves or nutritional muscular dystrophy. Works in tandem with vitamin E as well. Silicon. I call it the ultimate stress reliever. It not only works in the soil, it works in the plants. So outside the plant impacts the soil structure. Actually, high levels of soluble silicon, monosilicic acid, is ideal because it creates these silicon bridges or these structures that help with stress or with aeration. Also helps mitigate a lot of the salt problems that we have, okay, in some areas. It enhances fertility. Inside the plant, it affects the plant structure. Leaves are more erect. The leaves are a lot thicker because it lays itself along the epidermis. So when you have a thicker leaf, guess what? Piercing insects are less likely to go after that plant. Or fungus. Stimulates microbial growth, bacteria, fungi in the soil. In the soil, it also adds a protective layer around the roots. It's sort of like an armor. Helps with nematodes, helps with wireworms, helps with a lot of different issues. It helps regulate the uptake of heavy metals, regulates salt uptake as well. Increased resistance to biotic stress, fungal infections, take longer to spread, insects pest deterred. In fact, there's some studies even for herbivores that they won't go after that leaf because they don't like the sensation of that excess silicon in the leaf. So it will help you control herbivores as well. You heard me talk about abiotic stress, drought, excess moisture, mineral balance, and the list goes on. This is one of those use all silicons. Now, by any means, it's not the magic bullet, but you use it properly along with balanced nutrition, it will help you mitigate stress tremendously. This is some data. This is some silicon. 3 applications at different stages of growth, infrared planting, and then 2 foliar sprays, a total of 0.45 pounds per acre of actual silicon. This is monosylic acid, okay? You can see the control was very low with 3 sampling dates. Every time we applied some, we would test, and you can see it ranges from 400, 100 parts per million up to 8,000 parts per million. What it did is, sugar beets are notorious for leaf miner issues and for black bean aphids. We've eliminated all insect pressure, so we eliminated all insecticides. It also helps with nematodes. So on the soil applied, we've eliminated the use of fungicides and nematicides for that reason. Even though there is a high amount of nematodes, that silicon helps protect those roots as well. This is on wheat. That was a single foliar application, peaks 3 to 4, 32 ounces of a calcium silicate product, total of almost 0.3 pounds of actual silicon. Now, monocots are natural accumulators, okay? So you can see wheat can accumulate vast amounts, almost 5,000, but even when we applied it, it took us over 15,000 parts per million. We had no insect pressure whatsoever. Again, eliminated all insecticide applications. Potatoes are notorious for doing multiple fungicide applications in season. So we had strictly 4 foliar sprays throughout, starting at 30 days after emergence all the way to 70 days after emergence. Total of 0.3 pounds of actual silicon. You can see the control ranging from 200 to 700 parts per million, but every time we applied it, we were over 4,000 parts per million, the silicon and the sap. Eliminated all but 1 fungicide application with a seed. Eliminated all insecticides as well. We increase in tube production. All these were increased yields, increased quality, and reduced input cost. On this potato silicon project, we eliminated almost $250 an acre of input costs just by introducing silicon into the system.
Use selenium as well?
I'm sorry?
Use selenium as well?
On this particular product, no. It was just balanced nutrition with silicon on this. Yes, sir?
Do you have Colorado potato beetles?
Yes, we had, so in fact, during this study, we actually have late blight pressure around that particular field. We did not spray. We had a local consultant tell us, hey, there's some late blight, you guys better spray. And my farmer decided, hey, nope, we want to go full trial without a single fungicide application. We did not hit with any late blight whatsoever.
So they were on the plants, but you didn't see any damage, or they just...
No damage whatsoever with any late blight.
Well, what were the beetles doing? Or were they just kind of...
Well, we didn't get any insects whatsoever. They can't get it, they can't pierce that leaf. In fact, the farmer was surprised. They said, look at the leaf. You can actually strip the leaf apart because it was so thick and erect. And just, insects won't go after them. It's too much work. Insects go after survival of the fittest. They'll go for the weakest plants, okay? So when you've got a well-balanced nutrition plus the silicon as a protective layer, that is the ultimate way to protect your plants naturally. Yes, sir?
What product is available to remediate selenium deficiency?
There's several products out there. You might want to check with some products. I'm not sure, Biotech, Jake Biotech might have it. But again, you heard me talk about the sea salts. It's got some selenium in there as well. And that's probably where you're getting a lot of these benefits from these trace elements, selenium, cobalt, moly, vanadium, nickel. Some of these other trace elements that we're not accustomed to seeing, but they're in natural amounts in a lot of these products. Yes, sir?
What's the ideal number in plant sap for silicon, or is there a negative effect of too much silicon?
Oh, that's $1 million question. So the question is what is the ideal number for plants, and can you overdo it? There is not enough data. I've been doing the studies for the last 4 years using different products, calcium silicate, potassium silicate, sodium silicate, and it depends on the crop. And we're coming on with our own plant standards. Can we overdo it? We may, but to this point, I'm not seeing any detrimental effects with overuse of silicon. On the contrary, I'm seeing phenomenal results, not only in disease suppression, reduction of input cost, but quality. You know, fresh pack out on these buds was way over 95%. And we increased the production too by 15-20%, if I remember correctly. And every year we make tweaks, changes, and add different stages, and we keep increasing the production, okay? And these were Western varieties, okay? Which are phenomenal to only be yielding around 450 sacks per acre. We are pushing them now close to 700 sacks per acre. Just by managing nutrition, eliminating stress early on, and let the plant set its potential. We don't know what the genetic potential a lot of these crops are right now. Even the seed companies don't know, okay? Until you start working with nature, start eliminating stress early on, and then protect that plant all the way past reproduction stage, and that's how you can maximize genetic potential. That's how you can increase your yields while reducing input cost. So let me show you real quick here. Yes, sir?
Do you feel that would work with tomatoes?
I've got several tomato growers that
Have trouble with the seed bug?
Mhm.
I feel that would sound good.
I'm using it on all crops right now. I've got it on citrus. I'm doing some citrus trials. I'm doing some olive trials as well. I've done it on apples. I'm doing it on cherries. I've done it on tomatoes. It'll work on any crop because it will add that extra layer and thicker leaves to will protect them against any. Again, you've got to balance everything out. It is not a magic bullet by any means. But if you ask the plant and balance everything out, plus introduce silicon, it can do wonders for you. Okay? Yes?
Natural sources of silicon?
Natural sources, there are some natural mined products you can use, but how soluble is it in the soil? Okay? So the most soluble portions of silicate are stabilized products and come in a monosilicic acid form. Calcium silicate, potassium silicate, sodium silicate are the three sources that are extremely soluble. But they're only stable for about 2 years. So make sure when you're looking at these products, make sure they're pretty fresh and then use them, okay? Drawback is they're not very friendly. They don't mix well with other chemicals. So you might have to do an extra spray by itself to get the silicon or inject it by itself to be a benefit. That's the only drawback I have seen to date. Cost is some. Oh, not really. At the amounts we're applying, we're talking 20 ounces of a high pH soil with the water. Generally not an issue. Generally not an issue in the high pH. Question was, yes, pH of these silicone products is very high, over 11. We're only using 20 ounces, but you got to make sure that you have enough water. At least minimum of 15 gallons per acre. If you get any lower than that, like flying it on at 8 gallons per acre, you might have some issues. But generally, we have not seen any of those issues. Foliar sprays or water running it or injected. I'll be right with you. I just want to get these slides for you. And then, like I said, I'll be across. This is some, talking about getting your plant off to a fast start. Pop-up fertilizer, humic, fulvic, microbes. April 12th, taking 16, 4-day germination. That's what you want. That enhanced root growth, those lateral roots, okay? Organic wheat, planted September 18th. Use it half a ton of composted poultry litter, 1 pint per acre microbes. 8 days after emergence, look at that root system. Okay?
Back, please.
Hilled out corn, July 10th. 10 days later, we did a foliar spray. We did a silicon and a phosphorus. Recovered that plant. Sandstorm, 2015. Guy called me. It wiped out his corn early on. We did a foliar spray with silicon, phosphorus, humic acid. The following afternoon, he had a second sandstorm. Look at this, 22nd, completely recovered. Held out potatoes. This is one of our worst held out. This is in South Texas. These are some chippers that we work with, Frito-Lay. 30 minutes sustained of hells ranging from marble to golf ball size. It just completely annihilated our crops. So you can see that. 2 weeks later, we did a foliar spray. Completely recovered. Same time, watermelons, 1 week away from harvest, just totally annihilated the entire canopy and the existing March 29th. 2 weeks later, they're coming back. We did a foliar spray. 3 weeks later, harvested. Powdery mildew at harvest completely took out the canopy. We had to come out there and put a product like Surround to save the crop. Well, we came back, put a phosphite in this case, and we put some microbes in there. 3 weeks later, you couldn't tell they had been hit with powdery. So bottom line is, plant analysis, final judge overall of success or failure of your fertility program. You've got to ask the plant, make sure you ask the right questions, and make sure overall you know how to interpret it properly. Okay? There's my contact information. I'm not sure if I have enough time for questions. If not, I'll be in the consulting hall across through the hallway here, I believe.
We are going to ask everybody to take their questions across to the champagne room, which is just right across the hallway, and Noel will be over there as long as he wants to be, I guess. Thank you, guys.